A flowering plant grows in abandoned lots across every continent, producing compounds so powerful that German hospitals prescribe it officially, yet no pharmaceutical company can claim ownership. In 2013, researchers at the University of Freiburg published findings that shocked the medical establishment. This garden escapee demonstrated antibacterial activity against 13 different pathogens, including antibiotic-resistant strains. The study measured benzyl isothiocyanate concentrations reaching levels comparable to synthetic antibiotics. Yet this plant cannot be patented, cannot be monopolized, and refuses to submit to corporate control. It spreads through cracks in concrete, climbs chain-link fences, and produces seeds that remain viable for 5 years in soil. Modern medicine calls it Tropiolum majus. History remembers it as Nasturtium, the nose twister named for the peppery bite that signals its chemical warfare against bacteria. For 4 centuries it grew in monastery gardens as living pharmacy. Then antibiotics arrived, and the system demanded pills over petals. But Germany never forgot. The Commission E, Germany's regulatory body for herbal medicine, officially recognizes Nasturtium for respiratory and urinary tract infections. This is not folklore. This is institutional acknowledgement. If you value discovering plants the system tried to reduce to mere decoration, consider subscribing to learn more about botanical history the industry would prefer remained buried. The weapon Nasturtium deploys against bacterial invaders is glucotropiolin, a glucosinolate that fractures upon tissue damage. When cells rupture, whether from chewing, crushing, or bacterial invasion, an enzyme called myrosinase activates. This enzyme converts glucotropiolin into benzyl isothiocyanate, a volatile compound so reactive it disrupts bacterial cell membranes on contact. Research published in Phytomedicine demonstrated that benzyl isothiocyanate inhibits Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa. These are the bacteria responsible for urinary infections, wound infections, and pneumonia. The compound works by penetrating bacterial cells and interfering with protein synthesis. Unlike synthetic antibiotics that bacteria learn to resist through genetic mutation, isothiocyanates attack multiple cellular pathways simultaneously. Evolution cannot easily outmaneuver a molecule that disrupts 10 processes at once. The pharmaceutical industry understood this mechanism decades ago. They attempted to isolate, synthesize, and patent variations of benzyl isothiocyanate. But chemistry revealed an uncomfortable truth. The compound degrades rapidly outside living plant tissue. It cannot be stabilized for long-term storage. It cannot be concentrated without losing efficacy. The plant refuses commodification. This was not a failure of science, it was the triumph of complexity over reductionism. While American medicine dismissed Nasturtium as garden ornamental, Germany took a different path. In 1978, the German Federal Health Agency established Commission E to evaluate herbal medicines using the same rigor applied to pharmaceuticals. Nasturtium underwent clinical trials measuring urinary benzyl isothiocyanate concentrations after oral consumption. Results confirmed therapeutic levels reached the urinary tract within 90 minutes. Subsequent trials compared Nasturtium horseradish combinations against standard antibiotic treatment for acute sinusitis. The herbal preparation matched pharmaceutical efficacy while producing significantly fewer adverse effects. Commission E granted official approval in 1984. Today, German physicians prescribe standardized Nasturtium extracts as first-line treatment for uncomplicated urinary and respiratory infections. This represents institutional validation that no amount of marketing can fabricate. The plant succeeded where countless pharmaceutical candidates failed. It delivered consistent results without the side effects that plague synthetic antibiotics. Yet it remains classified as dietary supplement in the United States, denied the recognition it earned through decades of research. The distinction isn't botanical, it's economic. A plant anyone can grow in a window box threatens the business model built on prescription dependency. Pharmaceutical development follows a predictable formula: isolate active compound, modify molecular structure, file patent, manufacture exclusively. This model generated $1.4 trillion in global revenue in 2023. Nasturtium breaks every step of this formula. The active compounds exist naturally in dozens of related species: watercress, horseradish, mustard greens. Any attempt to patent benzyl isothiocyanate would be invalidated immediately, as prior art exists in botanical literature dating to 1597. The compounds themselves are too unstable for conventional pharmaceutical formulation. They require fresh plant material or freeze-dried preparations that maintain cellular integrity. This means no extended shelf life, no mass production in distant facilities, no centralized distribution. The plant demands proximity between grower and user. It refuses the global supply chain. Several major pharmaceutical companies attempted synthetic isothiocyanate analogs in the 1990s. Every iteration either lost antibacterial activity or gained unacceptable toxicity. The molecule's effectiveness depends on its natural context, the precise ratios of glucosinolates, myrosinase enzymes, and supporting phytochemicals that only intact plant cells provide. This is why Nasturtium remains free. Not because corporations are generous, but because nature designed it to resist commodification. Before 1928, when Alexander Fleming observed penicillin's effects on bacteria, Nasturtium served as frontline defense against infection. Jesuit missionaries introduced the plant to Europe from Peru in the 1570s. Within a generation, herbalists documented its use for scurvy, wound infections, and respiratory ailments. The 1653 Herbal by Nicholas Culpeper described preparations for putrid fevers and pestilential distempers. These weren't superstitions, they were accurate observations of antibacterial activity. During World War I, before antibiotics existed, military hospitals applied crushed nasturtium leaves to infected wounds. Medical journals from 1916 documented reduced suppuration and faster healing times. The practice continued until penicillin mass production rendered plant-based treatments obsolete in institutional settings. But obsolete to industry doesn't mean ineffective. It means unprofitable. A plant that grows from seed costs nothing after initial purchase. A pharmaceutical antibiotic requires prescription, insurance authorization, and repeat purchases. The economics ensured Nasturtium's demotion from medicine to garnish. We didn't abandon it because science proved it inferior. We abandoned it because capitalism demanded recurring revenue. In 2019, the CDC classified antibiotic-resistant infections as urgent threat, estimating 2.8 million cases annually in the United States alone. Methicillin-resistant Staphylococcus aureus, once confined to hospitals, now spreads through community settings. Carbapenem-resistant Enterobacteriaceae evade even last-resort antibiotics. The pipeline of new pharmaceutical antibiotics has slowed to near standstill, with only 43 new agents in development globally. Research costs for antibiotic development exceed $1 billion per drug. Return on investment remains negative, as effective antibiotics are prescribed sparingly and briefly. Pharmaceutical companies abandon the field as unprofitable. Meanwhile, Nasturtium's isothiocyanates remain effective against strains that resist conventional treatment. A 2018 study from the University of Southampton tested Nasturtium extracts against MRSA isolates. The compounds maintained bactericidal activity at concentrations achievable through dietary consumption. No resistance developed during 6 months of continuous exposure. The bacteria could not evolve defense against a molecule targeting multiple pathways simultaneously. This plant anticipated the resistance crisis 70 years before it arrived. Not through design, but through 400 million years of evolutionary refinement. The same chemical defenses that protected Nasturtium from herbivorous insects now protect humans from bacterial infection. Nasturtium operates as annual in temperate climates, perennial in frost-free zones. A single plant produces 5,100 seeds, each capable of germinating after 5 years in soil. Seeds require no stratification, no special treatment, merely contact with moist earth. Germination occurs within 7 to 10 days at temperatures above 55 degrees Celsius. The plant thrives in poor soil, requiring no fertilizer, no amendments, no intervention. Excess nitrogen actually reduces flower production and medicinal compound concentration. This is a plant that performs better through neglect than through care. It climbs using leaf petioles that curl around supports, reaching heights of 10 feet when provided vertical structure. Or it sprawls horizontally, forming dense mats that suppress competing vegetation. Every part contains isothiocyanates, leaves, flowers, seeds, and stems. The concentration peaks just before flowering when glucosinolate production reaches maximum. Harvest triggers branching, so the plant responds to cutting by producing more medicinal tissue. It grows faster than consumption depletes it. This is biological abundance that mocks scarcity-based economics. A 4 or 8 foot garden bed produces enough fresh material to supply one person's minor infection treatment needs for an entire growing season. Cultivation requires only seeds, soil, and space. Direct sow after last frost, spacing seeds 10 to 12 inches apart. Nasturtium establishes quickly, typically flowering within 45 days of germination. The plant attracts aphids preferentially, functioning as trap crop that protects neighboring vegetables. This dual purpose, medicine and pest management, demonstrates the integrated logic traditional gardens once employed. For seed saving, allow pods to mature until they turn tan and fall easily from the plant. Collect before full drying to prevent seed dispersal. Store in paper envelopes in cool, dark conditions. Viability persists for 5 years minimum, often longer. The seeds themselves contain isothiocyanates and were historically consumed as peppery substitute for capers. Growing this plant in 2026 is not nostalgia. It is practical preparation for a future where conventional antibiotics increasingly fail. It is botanical insurance against medical supply chain disruption. It is reclamation of agency in health decisions. Every seed planted represents rejection of pharmaceutical dependency. The system prefers you believe medicine must arrive in bottles, prescribed by authorities, purchased through approved channels. Nasturtium contradicts this entire framework. It demonstrates that effective compounds can grow in abandoned parking lots. It proves that patents are not prerequisites for potency. It reveals that the most valuable medicines might be those corporations cannot control. Germany's official recognition stands as evidence that institutional medicine can acknowledge botanical efficacy when freed from purely commercial incentives. The rest of the world watches, waiting for permission that will never come. Permission is not required. Seeds are legal, cultivation is legal, and knowledge is increasingly difficult to suppress. The revolution begins in a window box. It scales through shared seeds and documented results. It succeeds when enough people remember that before antibiotics were invented, plants like Nasturtium protected human health for millennia. This knowledge was never proven wrong. It was simply rendered unprofitable. Reclaiming it is not regression, it is correction.